So the plan for today is we need to finish these spinal control of movement slides, which means we need to talk about proprioceptors, and we need to talk about, there's a little bit on pattern generators at the end of this. And then hopefully we'll have time to at least have you guys talk about the homework for chapter 13, if not discuss at least the first few questions. We'll see how we do in terms of time. And I know as part of doing that chapter 13 homework, you already delved in a bit to muscle spindles, so that's good. So I hopefully don't need to explain every little thing, but I think that'll facilitate today's lecture. Also, just as a reminder, if you haven't already, meet with me about your outlines. What has happened is that, not saying people wait till the last minute, because it's not, it's the last week, but I did ask they be done, these outline meetings I think be done by the end of this week. However, my schedule is rather full as a result of this, so if you do not get in to meet with me this week, that is okay. Let's try to get in next week, though. I mean, the goal is that you meet earlier than like the day before you write the entire thing. So we don't want to wait until literally the last minute. So that's a very loose deadline, I think. But I have scheduled meetings with many of you over the next several days. And I've also, there's a few of you, I think I've told you, if you want to kind of like lurk outside my office during office hours, you're welcome to do so. But I literally got to the point that there was not a single open block of time over the next 3 days. And that's not a really good situation to be in, because like yesterday I didn't get a chance to eat lunch, and that starts to really suck. So in any case, I can't put any more people in the schedule, but if you would like to lurk outside of office hours, you may. But we can also meet next week, and it'll be fine, unless you really want to work on it this weekend. Any questions? Okay. Thank you for those of you that contacted me. I'm not mean to be. I want to meet with you. I really do. I want to meet with all of you. But there's 30 of you, and there's only so many minutes in the week. So I think where we left off is we were just starting to talk about proprioceptors. So, and in particular, we were diving into trying to understand how these muscle spindle organs work. And I think what we were talking about is that these muscle spindles are basically a collection of these skeletal muscle cells. They're called intrafusal fibers, intrafusal, so within fusal. This refers to them being within these muscle spindle structures, surrounded by a fibrous capsule with lots of nerve endings. And the job of these things is to be able to detect changes in length in the muscle once you've kind of set your muscle. So if I try to hold my muscle, my biceps muscle, such that my arm is oriented in this angle, and then you just sort of drop something onto it, especially if it was heavier than this, that might cause like a momentary change in length in that muscle, because I've sort of established a set point, but then if something unexpected lands on there, that could cause an unexpected increase in length of the muscle. The job of these muscle spindle organs is to detect changes like that. So when are there some sort of unexpected changes in length that are not driven by the overall contraction of the muscle? They're more sort of, once you've established a set point, what happens when you change length beyond that? So most of the sensory neurons are associated with this group 1A sensory axons. These axons that are coming out of here, that's these ones that are in red, these are gigantic axons. So the group 1A, you might remember back in the somatosensory chapter, we kind of had these different characterizations or different definitions for different kinds of axons. So mostly in that chapter, we were talking about like alpha B neurons are neurons coming from the skin, axons coming from the skin, that are pretty darn big, have a good amount of myelin, and they conduct signals really quickly. We did mention that there is this sort of theoretical A alpha axon that is pretty rare if it's coming from the skin, but if it was coming from the skin, we would call it an A alpha because they're a bit bigger than the A betas. They're larger axon diameter and even more heavily myelinated. That's pretty much what these axons are. These group 1A sensory axons are gigantic, massive neurons. The difference, though, we don't call them A alpha neurons because instead of coming from the skin, they come from muscles. And because of that, we use an entirely different classification scheme, which is frankly easier than the skin one. We just call them either group 1, group 2. The other ones would be called group 3 or group 4. These are group 1A, which means we're taking group 1 and we're dividing it into a 1A and 1B. And we do see the 1B sensory axons when we look at other kinds, well, when we look at Golgi tendon organs. So they're fitting, group 1 refers to this group 1. They're gigantic. These are some of the largest axons that exist in our body. And so as a result, they have very, very low internal resistance. They are very heavily myelinated, so they have massive membrane resistance. They are as instantaneous communication, as instantaneous communicators as you can get, as any human gets, is what we're seeing with these neurons. Okay? So their job, these guys, is that they are supposed to transmit information about stretch when we do have some increase in length in the muscle. So as we increase the length of the muscle, that'll cause this structure to get stretched. These sensory endings will pick up that stretch and transmit it almost instantaneously back up to the spinal cord. And ultimately back up to our brain, although a lot of what we'll talk about happens before you even get to the brain. Okay, so why are these things relevant? Well, the main place that we tend to experience these that is really clear and obvious is in the information that these sensory neurons, these proprioceptive neurons, as group 1A sensory fibers, send from the muscle spindle organs into our spinal cord that initiates what's called the myotactic reflex. And this is the reflex that we exploit when we start hitting each other with hammers in A&P lab, or you go to the doctor and the doctor starts hitting your knee with a hammer. What you're doing in that hammer, or with that hammer, is you're pounding in a specific spot on a tendon. So in this case, we're looking at the tendon that's associated with the quadriceps muscle. So the quadriceps is actually sort of complex. There's multiple muscles, rectus femoris, vastus lateralis, all those muscles that are all part of this, but they all converge on this same tendon. And then the tendon, the kneecap's sort of embedded in that, because it's really just a giant sesamoid bone. But then this little tendon right here, we're pounding in that spot right beneath the kneecap and right above the attachment point of this tendon. So in this case, what's happening is by pounding inward on that tendon, we are causing a momentary inflection where we're sort of pressing in on the tendon, just really briefly. It's while the hammer's colliding. But that's going to cause the tendon up here to jerk forward a little bit, which is going to cause a momentary increase in length in all of your various quadriceps muscles. Which means that these muscle spindle organs that are embedded in the quadriceps muscles are going to get a momentary stretch. You're going to have an increase in length in the muscle, which is going to cause a momentary tug on these muscle spindle organs. In response to that tug, these group 1A sensory axons are going to send signals as they detect the stretch, they'll send signals from the muscle spindle up the 1A afferent axon. It's a sensory neuron, so it goes up through the dorsal root of the spinal nerve. There's that dorsal root ganglion up here, because these are unipolar neurons. And then that will send a signal then into the spinal cord. And then here, they're kind of showing it. I'm not sure this is quite accurate. They're showing this axon going all the way down and hitting an alpha motor neuron. I think sometimes there's some interneurons that are involved too. But the point is that we're immediately getting a synapse that's more or less sending information directly from this 1A axon straight to an alpha motor neuron. That alpha motor neuron then leads directly back to those quadriceps muscles. So the same muscle that housed the muscle spindle organ is now being stimulated to contract by this alpha motor neuron via this reflex loop. So the result of this is when somebody hits you in the quadriceps tendon with a hammer, that'll cause a reflex loop that's going to cause your quadriceps organ to contract briefly, and it results in a little twitch, right? And we've all experienced that. And in lab we do the same thing. You can hit right here in your Achilles tendon, and it does the same thing. It sort of triggers a little reaction. That's actually, I don't know if you're one of the people that your leg kind of twitches. Mine does this, if you're sitting there, sometimes when I'm concentrating, this seems to be what happens, that my leg will just start like vibrating. And I think what's happening there is I'm basically repeatedly stretching my Achilles tendon, which stretches some muscle spindles in my gastrocnemius muscle, which causes it to contract, and I'm sort of repeatedly causing this little reflex loop to fire. But that's a manifestation of the ability of these muscle spindles to detect small little changes in length. And when they do so, the reflex loop, the myotactic reflex loop, causes that same muscle that the muscle spindle is attached to contract in opposition to that stretch. which, when you're sitting on an exam table and you're just seeing your leg twitch like that, doesn't really make a whole lot of sense why that would be a valuable thing. But if you think about it, like what this thing would normally do in your leg is that, so here's my example. So if I'm walking along, right, and I sort of set my leg as I'm walking forward and I step forward, and maybe because I'm walking outside in the grass and I'm not looking down, maybe my foot goes a little bit further, or there's like a little bit of a depression. So I kind of lean forward a little bit more, and it takes me a second more to land, so I hit with more force than I'm expecting. We've all done this, right? Maybe it's just me, but where you have, you're sort of momentarily a little bit you have this loss of coordination because the ground wasn't where you expected it to be. What will happen, though, is as you start to lurch forward and you're starting to fall forward on your face, your quadriceps muscles are engaged, and they're going to experience a little extra stretch, a little extra momentary change in length as you're kind of falling forward in response to this. And so what will this reflex do as you start to stretch your quadriceps muscles? What is this going to do for you? I mean, what do you think? So I'm leaning forward like, oh God, I'm starting to fall, but I haven't even realized it yet because it hasn't even made its way to my brain. What's the result of this reflex loop going to be on my quadriceps muscle? Yeah. So I'm lurching forward. I'm starting to have a bit of an unexpected change in length in my muscle. That'll stretch the spindle, which will trigger the 1A axon to fire, send a signal then to my alpha motor neuron. These alpha motor neurons will send signals back to my quadriceps, and I will increase contraction in this quadriceps muscle to oppose that increase, that unexpected increase in length that I'm experiencing. This is all reflexive. Like, this is not something where your brain has to be aware that you're falling forward, and you have to make a decision like, gee, I think I probably should add a little bit of contractile force to my quadriceps muscle to keep myself from falling forward. We are immediately, via this reflex, probably before the signal even gets to our brain, already contracting our quadriceps muscle to help correct for that change in length of the muscle and stabilizing, making sure that we're applying appropriate amounts of stimulus to the muscle to get it to contract enough to oppose that sort of increase in length. Does that make sense? Like more or less what it's doing? That to me is like the best example of why this would be valuable in real life as opposed to sitting on an exam table, is that this is going to allow you to immediately correct for unexpected changes in muscle length by just contracting a little bit harder via this reflex loop. And it's all, reflexive. It's super fast, and it doesn't require brain input. Okay? This slide's showing kind of that, or kind of the action of the reflex loop over time, just so we can sort of visualize it a little bit better. So here the idea is, here this row is going to show the load that's applied onto a muscle. This is going to cause a momentary increase in length of the muscle, which is going to be shown here. This is observing what's happening from the 1A sensory neuron that's coming out of the muscle spindle. And then the last row is showing the response of the alpha motor neuron. So what you see is they're starting with a situation where the muscle is basically just suspended here. If an additional amount of weight or force is added to the muscle, that's going to cause the muscle to increase in length momentarily, just a little bit. But when it does so, as the muscle becomes a little bit longer, that's going to cause stimulation of the spindle apparatuses that are present within that muscle. And they're going to start firing signals up this 1A axon. So you see, initially you're sort of at your set point. And then you have this momentary increase in length of the muscle that's caused by the weight. That causes the sensory axons to start firing like crazy. And then that in turn, those signals travel up, oops, I went backwards. Those signals travel up to the spinal cord, innervate the motor neurons in green. The motor neuron is shown down here. And so within a moment of the 1A axons starting to fire, you see the motor neurons start to fire in response, and now we're contracting that same muscle. So we're opposing that change in length. And because of that contraction, now the muscle is going to get back to its normal set length. So it'll shorten now in response to the signal, because you're increasing the stimulation to contract the muscle. So any questions on that reflex? Is that making sense? There's one other aspect to this, which is kind of cool. And that is something we see in a lot of these different reflex arcs, and we see it again on the ones that come up on the homework, is that we also, in addition to stimulating a particular muscle to contract, we will simultaneously have what's called reciprocal inhibition of the antagonists, which is a great mouthful, but all it's saying is that reciprocal would sort of means at the same time or in exchange for, we're inhibiting antagonist muscles. So these are the muscles that would cause us to move in the opposite direction. So that's illustrated in this figure. They went back to the arm for this figure, so we'll do the same. So in this case, you see, I'm going to start with repeating what we had in the prior slide. Muscle spindle gets excited because there's been some momentary increase in the stretch, which is causing a brief increase in the length of this muscle organ. So the muscle spindle gets excited, sends signals up the 1A afferent axon, and then that in turn causes stimulation of the alpha motor neurons that lead back to that same muscle, and they'll cause that muscle to contract in response. So all that's exactly the same. There's one additional wrinkle that this figure shows, though, and that is that in addition to stimulating the same muscle that the muscle spindle is housed in, there's also stimulation of an interneuron. In this case, they label it as an inhibitory interneuron. So we stimulate the inhibitory interneuron. This, in turn, will inhibit this other motor neuron. You say, well, why would we do that? Well, look at where this motor neuron goes. This motor neuron leads not back to the biceps, but in this case, it's leading to the triceps. So if you think about these muscles, biceps causes flexion of your elbow. Triceps causes elongation. So these are what we call antagonists to each other. The triceps antagonizes the biceps, and vice versa. So whenever the triceps is contracting, that's going to cause extension of your elbow. Whereas whenever the biceps is contracting, that'll cause flexion of the elbow. So what we're doing is at the same time that we're stimulating the biceps here to contract, we're simultaneously inhibiting the motor neurons that lead to the triceps. So we're going to force them to relax in response to this stimulation. So why would we do that? Like, why do you want the triceps to relax at that moment? What do you think? So, I mean, we know we're trying to, the biceps is getting too long, so we're trying to contract and get the biceps back to its normal length. So why would we want to shut down the triceps at the same time we're stimulating the biceps? What do you think? Yeah, so they're set up so that they are always antagonizing each other. So whenever the triceps is working, it's basically undoing what the biceps is doing. So if you stimulate, if they're both running, I mean, you're basically just going to kind of cause your arm to seize up then, right? If both the triceps and the biceps are contracting simultaneously. So what we do is that in order to cause the biceps to be able to cause the elbow to flex a little bit in response to that muscle spindle, we will simultaneously shut down the triceps so that these guys stop contracting. And that's going to allow the muscle spindle, well, this myotactic reflex, to get the elbow to shorten the length of the biceps and get back to our set point. So right, so we don't want them fighting each other, right? We want to shut down the triceps whenever we're trying to activate and stimulate the biceps. That make sense? All right. So that's one kind of proprioceptor. That's the main function that we'll talk about it doing. Please also be aware, though, I mean, it's showing these two axon collaterals. There are other axon collaterals that will allow information to be sent up into our brain. And so our brain is receiving information from these proprioceptors even as they're feeding into these reflexes, so that we are aware of what our muscles are doing and any stretches or changes in length that are being placed upon them. This is part of how we're sort of aware of what our limbs are doing even when we're not looking at them. So in the case of the disembodied woman, which we read about for the somatosensory chapter homework, I think we would expect that some of this input is not getting its way up to the brain. She might still have the spinal reflexes, but at least the information is not getting up to her brain, which is she's, as a result, going to lose her ability to kind of have a sense of her body and what her limbs are doing in her body. Here's another kind of proprioceptor. This is called the Golgi tendon organ. These things also are found associated with most muscles. And these things also kind of are stretch receptors in sort of a similar kind of a way to what the muscle spindles were doing. I think the difference here is I tend to think of these things as kind of like a warning device. I describe them as a strain gauge. They're monitoring the tension that's placed on the muscle through the tendon. So the tendon, as the tendon gets stretched during a contraction or when you're trying to actually do some kind of work with the muscle, that stretch is being monitored by these Golgi tendon organs. So it's not really interested in how long the muscle is, which is what the muscle spindle is all about doing. It's just basically monitoring how much tension is being placed on this structure. And that can, I think the easiest way to think about what these things do is that, and we had a question, I think from you last time, about what happens if you're generating tons and tons of force. Could you actually cause damage to the tendon? That's really what these things are designed to help you avoid, because as your tendon starts to be stretched and stretched and stretched, these Golgi tendon organs will detect that stretch and send a signal up, in this case a 1B axon. So these are group 1B axons. They're not 1A axons. That means they're still in the group 1 category of axons, but they're slightly smaller and have slightly less myelin than the 1A fibers. So they don't send signals quite as quickly, but they're super duper fast. I mean, they're still faster than touch receptor axons, which were those A beta axons. These are super fast. When you start to place additional stretch on the tendon, the Golgi tendon organ has sensory endings that can detect that stretch, and that will then start to send signals up the 1B axon. And the result of that is that it stimulates a different kind of a reflex loop that, well, let's just walk through the reflex loop. There's a point on that last slide I'll come back to in a sec. So here is your Golgi tendon organ. It's found at the end of most muscles, embedded in the tendon. You have 1B axons that when there's a lot of tension that's being placed on this tendon, that 1B axon will send action potentials up into the spinal cord. And in this case, the main result of that is that the 1B axons stimulate an inhibitory interneuron. This would be a different one than what we saw in the prior picture, but it's another one that's housed within the spinal cord, which would then shut down or inhibit this alpha motor neuron. And let's look where this alpha motor neuron goes. It goes to the same muscle that's associated with this tendon. So the result is that if you stretch the tendon substantially, that's going to cause substantial input from the Golgi tendon organ, which in turn is going to cause inhibition to be sent down to this muscle. So you're basically turning off the muscle in response to this stretch. So to me, like, the easiest way to think about what these things are doing, and it's not the only function they have, but the easiest, most straightforward function is that if this stretch gets to be dangerous to the point that you're starting to come close to getting towards a point that you could damage the tendon and cause the tendon to cleave or tear off the bone, the Golgi tendon organ would sort of alert you to that, and in response it would trigger this reflex loop that will shut down contraction of the muscle, hopefully before any damage is done. So you're kind of, it's kind of a safety valve here. We're saying, oh no, we're about to break, so let's shut down this muscle. It will relax, and by relaxing this muscle, we should relieve the stretch that's been placed on that Golgi tendon organ. Is that okay? Makes sense? So that's what I'm saying here. 1B axon works through an interneuron to inhibit the alpha motor neuron, and it's that same muscle that's associated with this tendon that's being inhibited now. It's not the antagonist, just sort of the opposite of what we saw before. Under extreme tension, that's going to protect against damage. It'll prevent you from hopefully contracting so hard that you tear your tendon off the bone or you rip your tendon in half. Yeah?
So I always get these two so confused. I have no idea why, but so basically the muscle fibers are using the myotactic reflex, and then the Golgi tendon organ is using the reverse myotactic reflex.
Yeah, this is the reverse myotactic reflex.
Okay, and so therefore, since this one is causing it to relax,
Mm-hmm.
and the muscle spindles, or the muscle spindles are causing it to contract.
Right.
Okay, and so my
Everything you said is correct. So it's very much the case. These seem like it seems like the Golgi tendon organ should be fighting against the spindle apparatus all the time. There are a few differences in terms of what they're doing. I mean, one is that in terms of total shutdown of the muscle, the Golgi tendon organ will only do that under really severe tension. So, this isn't going to be something that's going to be firing all the time and causing your muscles to just go flaccid and negating any input that's coming from the spindle apparatus. So it's sort of like only under severe stretch do you get to that level. The other difference has to do with like what specifically they're detecting, which actually is coming back to this point. So I said I would come back to this point, this figure here. So this is a prior slide. Sorry, I kind of skipped over it, but it was not in my train of thought at that moment. And what this is trying to show is that the muscle spindle organs versus the Golgi tendon organs are kind of picking up different aspects of stretch that could be placed across a muscle. So in the case of a Golgi tendon organs are found in that tendon that's at the very end of the organ. And so as a result, anytime there's any kind of force that's placed on the muscle, it's going to be responsive to that. And that includes when the muscle is contracting. Even contraction of the muscle is going to cause stretch here, but also if there's some sort of inadvertent force that's placed on the muscle, it's also going to pick up that kind of force. And that's because we would say that it's in series with the main body of the muscle. The muscle spindle organ, in contrast to that, is kind of separate. I mean, it's embedded within the muscle itself, but it's connected by connective tissue all the way to the two attachments, the origin and the insertion of whatever muscle you're looking at. And so contraction of the muscle itself doesn't actually cause much stimulation in the muscle spindle. It would only be sort of outside forces that the muscle spindle will be responsive to. That sort of unexpected additional amount of force that's being placed on a muscle once you've sort of established its set length. So I guess all this is to say that they look like they would be fighting each other, and on some level maybe they kind of are, but most of the time they're responding to different kinds of things, and they're certainly performing different tasks for us. Muscle spindles are all about helping us to maintain the desired set point of our muscles. So anytime we have an increase in length, the reflex loop causes that the myotactic reflex will cause the muscle to get back to its original set length. Whereas, again, I tend to think of Golgi tendon organs as like the safety valve. So when you're about to cause damage, it shuts down that muscle before any damage occurs. So I hope that kind of makes sense. They're sort of doing two different things. Now there's another thing about the Golgi tendon organs. It's not strictly that. They also have this function of just kind of smoothing out contractions as well. So you could sort of imagine if you have, we talked about ways in which you can have these sort of graded contractions, but at a sort of a whole muscle organ level. But one thing that will happen is that if you have sort of a sudden jerk of a contraction that's occurring within a muscle, which can sometimes occur, that'll cause sort of a brief stimulation of this Golgi tendon organ as you're contracting. And so if the muscle isn't moving really smoothly through its contraction, it's starting to jerk a little bit, the Golgi tendon organ will pick up that jerk. And through this very fast reflex arc, it can cause a really brief minor inhibition, not a shutdown, but just like a minor depression of the contractile force to start to smooth out those contractions. So another function of Golgi tendon organs, beyond just sort of being a safety valve, is they help to kind of smooth out contractions of your muscles. They're not going to shut them down altogether most of the time, but they help sort of get away from sort of spikes of contractile force where they kind of dampen that out. This is really important when you're doing really fine motor control. So when you're finally trying to move your fingers or your arm or your hand and you're trying to do something really precise, in that circumstance, you don't want substantial force of contraction, right? I mean, that's not the goal. We're not trying to move mountains here when you're manipulating a pencil and trying to write with a pencil. What you're trying to do is make really precise sorts of actions. And so you don't want jerky types of actions that are powered by strong force. You want to dampen those down. So under those circumstances, these Golgi tendon organs can help you to perform nice, smooth, non-jerky types of actions. Last point on proprioceptors. I know you probably have heard about Golgi tendon organs. You've probably heard about muscle spindles before. This is maybe some review, but hopefully a little bit clearer understanding. It's clearer than I usually present in an A&P, at least, when I teach about these things, as far as what the roles of these different organs are to do. There are other kinds of proprioceptors that are present within ligaments. They're present within joints. Most of these are mechanoreceptors. They're responsible for detecting and picking up any kind of change in the stretch, but that's going to result in input that's going to be sent up into your spinal cord and ultimately up into your brain to help detect movement of limbs, the current angle of limbs. That's what proprioceptors are really all about. So any questions on proprioceptors? All right, last couple slides. Motor programs. So we do have, I want to talk about a couple different types of pattern generators, but I think I started the chapter by talking about the idea that your spinal cord does have the ability, it has little circuits that can generate patterns of motion. So for example, on a basic sense, some of our typical walking motions, which is sort of this repeated extension, flexion, extension, flexion of our two legs simultaneously, those are on some level powered by little pattern generators that are housed within the spinal cord, and then we can modify those with descending input or output that's coming out of our brain. So what the end of this chapter is doing is talking about single cell pattern generators as well as pattern generator circuits. And some of this stuff very much harkens back, I think, to the stuff we were talking about in chapter 5, and also that showed up in this last exam about simple circuits, the Tritonia escape response, this kind of thing. So the idea is that we have many motor programs that are created by pattern generators that exist in the spinal cord. And these can be single cell pattern generators as well as sort of more complicated circuit pattern generators. So I don't have a good example of a pattern generator from humans, although I think we do have some. I'd have to like really sit down and think about if I have a good example of these. But one of the best studied is this lamprey swimming cell. And this is described in your book. So the idea here is that this is a single cell that exists within lampreys that helps to understand how you can have sort of alternating, if you've ever seen a lamprey, lampreys are these fish, right? These jawless fish. And they swim in this really nice, smooth, undulating kind of a motion. They look a little bit like a sea snake or something as they swim underwater. It turns out that that pattern of alternating sort of contraction, relaxation, contraction, relaxation, contraction, relaxation is powered in large part by these swimming cells, which are neurons that have this, by themselves, they generate sort of an alternating period of excitation and then relaxation. And this is basically how they work. To get the swimming cell started, you do have to send external input to it. So in this case, what they're showing is that you'll have glutamate being released onto these swimming cells. There's the glutamate molecule right there. And it binds to a glutamate receptor, which is this NMDA receptor. There's actually several different kinds of glutamate receptors. This is just one kind, okay? But when glutamate binds to its NMDA receptor, that causes this channel to open, and this is a cation channel that allows both sodium and calcium ions to flow into the interior of the cell. So as they do that, what's that going to do to the membrane potential, do you think? Yeah, it's sodium, potassium coming in, or sorry, not potassium, sodium, calcium coming in. More positive, more negative, what do you think? More positive, right? Yeah, we have positively charged ions flowing in. So that's going to cause the membrane potential to depolarize, and that's what you're seeing here. So the membrane, in response to glutamate binding, membrane depolarizes within the swimming cell, and then at this point, you get up to threshold, and it fires a series of action potentials. But then a cool, weird thing happens. The calcium that came in with the glutamate does what calcium often does, is it functions as an intracellular signal. That calcium comes over and interacts with a potassium channel, which opens this potassium channel. And it's not instantaneous. There's a few milliseconds that go by before this happens. But as a result of opening the potassium channel, now we've got this huge excess stream of potassium leaving the cell, which is going to cause inhibition, right? We've got positively charged ions leaving, and that's going to cause the membrane to get more negative. And so that's what's happening here. So the membrane gets more negative, and at the same time that that happens, you also have magnesium comes along, and there's a low concentration of magnesium outside these cells, but magnesium comes in and basically functions as a cork that blocks, it interacts with components of the this sodium-calcium channel and blocks the opening to that channel. So at the same time that we open potassium channels, we also block the calcium-sodium channels that we just had. And now, so the only current that's moving across here are these potassium ions moving across. So as a result, the membrane gets very negative after, once this occurs. So by opening the channel, we depolarize, fired some action potentials, and then very quickly we sort of reset our situation until we get to a negative membrane potential. Well, I'll point out, glutamate's still bound here, right? We still have neurotransmitter bound to this channel. We're still technically stimulating it, but at this point now we sort of become negatively, we've seen the resting membrane potential drop. And then what happens is now calcium's no longer coming in. So we don't have as much calcium around the potassium channel, so it closes. When the potassium channel closes, then the membrane potential, oh my goodness, excuse me, the membrane potential can drift upward. And as a result, you'll have manganese sort of popping out, no longer being in the way, which will cause calcium and sodium to start to rush in, which again will cause a depolarization. And again, all the while, glutamate is bound all through this time. So the cell by itself, through the interaction of these two channels and the ions that are involved, will periodically depolarize and fire some action potentials, and then hyperpolarize, and then depolarize, hyperpolarize, depolarize, hyperpolarize, all by itself, even though it's constantly under stimulation from glutamate this whole time. It's cool, right? I mean, this is a really weird sort of scenario. We're used to seeing, neurotransmitter binds, so the channel opens, and as long as the neurotransmitter is around, we keep having the channel being open. But that's not the case in this situation. You've got this sort of interactive effect between these two channels that compete with each other as long as glutamate is present. This process will finally stop when glutamate detaches from the receptor, at which point the cell is going to drift back down to its resting membrane potential and be nice and stable because both these channels will be closed. Okay? So again, I think the thing that's cool about this is we don't have to have a circuit. We have one cell that has competing channels, and those channels result in this one cell turning itself on and off and on and off and on and off in turn. All right, so that's one way you can have a pattern generator, is by something analogous to the lamprey swimming cell. And there's other examples of these. It's just this is the best characterized of them. But then many motor patterns involve much larger circuits. So guys, remember this. This is the Tritonia escape response. So I'm not going to go through the whole thing again. But we saw how this circuit resulted in this alternating pattern of excitation and then inhibition of a particular muscle group. And it even resulted in sort of, a reciprocal inhibition process where the dorsal flexion neurons would inhibit the ventral flexion neurons and vice versa, such that you only got one going at the same time. But it resulted in this nice motor pattern sequence occurring. So there is another example in your book that shows something like this. I think we get back to that in homework. So I'm going to stop talking at that moment. So what I'd like to do is we've got 12 minutes left in the class, and I would like to turn this back to you and have you start going through the homework. And we'll see if we have a little bit of time at the end. We can maybe at least talk about the ALS question, which is the first question. But give it a chance to talk it out yourselves, and then we'll talk about them in detail on Friday. Any groups? Go for it.
So the first principal mechanism was due to excitotoxicity, due to overstimulation by glutamate, which could essentially kill the normal alpha motor neuron. Second, in the same type of thing, there's also a defective glutamate transporter, which leads to increased exposure of active neurons to glutamate, which would result in death. And then the second principal mechanism would be due to a genetic mutation. One type of gene mutation leads to defective superoxide dismutase, which causes the production of a superoxide radical, which leads to an irreversible cellular damage. and this could be due to malfunction of those cells.
What was the last thing you said? The malfunction of
Glial cells.
glial cells. Anybody put anything else or wanted to add anything to these? I'm misspelling all my words today. They make you want to go get some cycad nuts. Did you see that thing? So there's this group of people on this island that were eating cycad nuts, and there's apparently something in there that was an amino acid. Glutamate is an amino acid. But there was something in these cycads that they were consuming that was mimicking glutamate. I'm not really clear exactly what that is, if it was just straight up they're getting too much glutamate, or if it was something related to glutamate that was causing stimulation of these same receptors. But the result is that these people were getting conditions that looked a lot like ALS, where they were losing motor control. And apparently it was due to this thing they're getting from their diet. That's usually not what happens. That's one case where you've had something like ALS occurring. Usually this is caused by something happening that's resulting in overstimulation by glutamate. And so one specific mechanism that was talked about in there was you can have, I think this can also be a genetic-based mutation in the transporter protein. So if you're losing your transporter, you no longer have this way to clear glutamate out of synapses after it's been released. At that point, then it can overstimulate these alpha motor neurons and result in them basically just perishing due to the overstimulation. The other thing it mentioned is superoxide dismutase, which seems like a very different mechanism. It is a very different mechanism. So there you're dealing with an enzyme. The enzyme is designed to eliminate superoxide particles, superoxide radicals, which is basically oxygen with extra electrons attached to it. And those in turn can result in damage occurring, where if you've got too much of these superoxides around because you no longer have a functional superoxide dismutase which is supposed to destroy them, then you're losing the ability to control your oxidative damage, runs rampant, and ends up causing these same cells to die. All right, so I'm out of time, so we will start.